Okay, so today we're going to talk about the overall function of the kidney. And we're going to start by talking about ultrafiltration. So ultrafiltration takes place in the glomerulus where it interacts with Bowman's capsule. So as blood comes into the glomerulus, there's a really high pressure, right, as it hits a dead end. And the way you can think about this is if you've ever taken a hose and put your thumb over the top, builds up the pressure of the water, tries to shoot out around it. Same sort of thing happening here. As the water shoots in, hits the dead end of the glomerulus, and hits really, really high pressure buildup. And what that's going to do is it's going to try to allow that liquid to seep out somewhere. The question is, where is it going to go? And we need to look at it a little bit more microscopically. So if I draw the capillary in the glomerulus, the wall of the capillary is made of cells. And there's actually tiny gaps in the wall. So here's the gaps. And on the other side, here's the wall of Bowman's capsule. This is Bowman's capsule. And there's gaps in those walls as well. And so when the pressure builds up, what's going to happen, right? We got our blood cells running through here. What's going to happen is the pressure builds up in this capillary. The liquid's going to want to seep across out of those holes, kind of like water spewing out around your thumb in a hose, and go into Bowman's capsule, right? And so this is how filtration will occur. But think of filtration. You have a filter. In this case, there's what's called the basement membrane. And it's not actually a membrane so much as it is like a sort of like a mesh, right? A mesh of fibers. We call it the basement membrane. And kind of like a coffee filter, it's going to control what can come in, what cannot come in. So pretty small molecules will be able to pass through, but no large proteins, no cells will be able to get to the other side. And so what eventually gets to the other side, the term for that is the glomerular filtrate. The glomerular filtrate is what gets to the other side. So let's look quickly at different molecule types. So will they pass through? Will glucose pass through? Yes, glucose is very small. It can pass through the basement membrane. Will amino acids pass through? Yep, amino acids are really small. They can pass through the basement membrane. What about sodium ions? Again, yes, very small. And same thing for other ions. So things like Cl- or K+, or calcium ions. All those can pass through. Can water pass through the basement membrane? Yes. Can urea pass through the basement membrane? Yes. Again, also very small. Now notice, right, when you talked about membranes in biology class, things that were charged, like sodium ions, couldn't pass through. This is not a phospholipid bilayer. This is more like a really fine mesh. So the basement membrane is something different. But now we get to larger things like proteins. Proteins are too big. They can't pass through. Red blood cells, way too big, can't pass through. White blood cells, way too big, right, can't pass through. And so when you have blood in your urine, what it's like if you've made coffee and the coffee filter is broken, right, it's ripped and you get coffee grounds into the coffee. That's the same way when you have blood in your urine, it's usually a big concern. There's something wrong with your kidney, right, that there's some sort of rip in the basement membrane allowing those red blood cells to escape. Okay, so that's at the Bowman's capsule and the glomerulus. Then we get to the proximal convoluted tubule. So we'll call it the PCT, proximal convoluted tubule. And we're going to do something called selective reabsorption. So selective means certain things. Reabsorption means they're going back into the bloodstream. So why do we need to reabsorb? Well, ultrafiltration just takes everything away. We'll get, if we go back, right? we'll have, say, something that can pass through like a sodium ion or an amino acid, we'll end up with exactly the same concentration in the blood and in the filtrate. Maybe we want to save those amino acids, right? Or save all that glucose. We want to pull that now back in, but leave the urea out. We don't want to save that. We can go and get rid of that. So we've got our proximal convoluted tubule. So here's like the wall of the proximal convoluted tubule. Again, it's made of particular cells. So here's our PCT. And we're going to want to specifically move certain things from the proximal convoluted tubule back into the bloodstream. And so to move these really specific things, right, so say glucose, we want to move glucose out, or we want to move amino acids out. In order to do that and move those and move all of them out, right, so even going from a low concentration to high concentration, we're going to use active transport. So we're going to go through some sort of protein, right, and we're going to use ATP to do that. Right, use ATP. This one uses ATP. And so the glucose and the amino acids will get pumped by active transport, right, from the inside of the PCT into the wall of the PCT, and eventually they'll go back into the blood. So by doing this, we're able to remove almost all glucose and almost all amino acids from the filtrate. You generally don't find glucose or amino acids in your urine. If you do, it's because you're sick. So, but then the question is, what does it do to water? So if we're moving all of these solutes, glucose and amino acids, out of the PCT back into the blood and the area around it, what does that do to the water potential? Well, having all those solutes is going to lower the water potential out there. And then what does it do to the water potential inside the PCT? Well, we're losing solutes, so we now have a higher concentration of water, which means you've got a higher water potential in the PCT. And so what this means is that water is going to move by osmosis from a high water potential to a low water potential. And in doing this, about 65% of water is recovered here. If we didn't have this process, we would lose so much water in our urine that we could never stay well enough hydrated. So here we recover about 65% of our water. And then it gets even better in terms of water recovery in the loop of Henle. So the loop of Henle, we draw it. So we've learned about, there's different parts of the kidney, the cortex and the medulla. And the proximal convoluted tubule runs through the cortex, but then the loop of Henle dips down into the medulla. Now, to understand how the loop of Henle works, we need to think about it in two halves. So on the left side, so liquid's coming in this way, coming out this way. So here it's going down, so we call this the descending limb of the loop of Henle. And over here, we call this side the ascending limb. And it's actually easier for us to think about the ascending limb of the loop of Henle first. So ascending limb is special. I'm going to kind of draw a thick line on the side here. And the reason I'm kind of drawing this thick line is to represent the ascending limb is impermeable to water. However, it is permeable to ions, particularly sodium and chloride ions. And so what's going to happen in the ascending limb is that sodium ions and chloride ions are going to be actively pumped out from the loop of Henle into the medulla. And it's active, so they're using ATP. And so what that's going to do is it's going to mean the medulla is full of sodium and chloride ions, right? So we got, lots of sodium ions everywhere. And lots of chloride ions everywhere. Now the reason this is so important is because of what it does to water potential. So when you move all of those sodium and chloride ions into the medulla, what they do is they give the medulla a very low water potential. So I'll kind of write low water potential all over here in the medulla to remind us of that. And so now when we go back to the descending limb, as the liquid is flowing through here, the descending limb is permeable to water. That means water can pass through it. And so what's going to happen is that water, because there's a higher water potential in the loop of Henle and a lower water potential outside of the loop of Henle, water is going to move out of the loop of Henle. And this is going to allow even more water to be recovered than we are able to get in the proximal convoluted tubule during selective reabsorption. Now, you might ask yourself, why are we doing this two ways? Like in selective reabsorption, we first move solute and then water followed. In loop of Henle, again, we use active transport to move solute and then the water follows. Why don't we just move the water directly by active transport? And this will be kind of one of the themes that a sentence you'll hear me say over and over again. Water can never move by active transport. If you figure out how to do it, you can probably win a Nobel Prize. Water can't move by active transport directly. So in order to move water, we have to move other things, move solute, so that we change the water potential and water will follow. The final thing I want to say is we're going to come back to the medulla in our next lesson, because having all of this salt in the medulla with a really low water potential has become really important for what happens in the collecting duct in osmoregulation.